Hello everyone. InformationBoxTicket Lifestyle brings you Biotechnology Unit 4 Plant Biotechnology. But before that, don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my lectures. First, we will look into the table content of plant biotechnology. We'll go through introduction, conventional plant breeding versus molecular plant breeding, transgenic plants, gene transfer methods in plants which are vector-mediated or indirect gene transfer, which includes mechanism of gene transfer by Agrobacterium. Number 2, direct gene transfer, which is physical methods of gene transfer, which includes microinjection, chemical-mediated gene transfer, electroporation, biolystic method, Chloroplast engineering. Then we will head to applications of transgenic technologies in plants, which are herbicide tolerance, insect resistance, disease resistance, abiotic stress tolerance, delayed ripening fruits and vegetables, male sterility, improvement of nutrient qualities of plants, transgenic plants as bioreactors, which is molecular biofarming, biodegradable plastics, plant secondary products. And lastly, we will look into risk associated with genetically modified food and oversight into it. So let's start with the introduction of plant biotechnology. The recent decade has seen extraordinary progress in plant biotechnology, from the study of the science to large-scale commercial applications. Plant biotechnology applications may be classified into two categories. Number 1, those aimed at traditional plant breeding, example, better yield, quality, resistance to pests and diseases, tolerance to abiotic stressors, and number 2, those aimed at wholly unique applications such as use of plants as bioreactor to generate pharmaceuticals, vaccines, or biodegradable plastics. So the difference between conventional plant breeding versus molecular plant breeding is plant breeding is founded on Mendelian genetics principle. In past, plant breeding was considered an art, with the selection of superior genotypes of a given crop relying heavily on subjective judgments made by the breeder. With increasing knowledge of the genes underlying useful traits, plant breeding has become a more directed and scientific activity. As a result of the generation of molecular maps of crop genomes, extensive sequencing of express sequences and genomic sequences, and the study of genome organization, repetitive and non-coding sequences, and the ability to identify polymorphisms at a specific loci that can be exploited as molecular markers. Transgenic plants. Transgenic plants are plants that have foreign DNA added to them. There are three basic motivations for generating transgenic plants. First, the inclusion of a gene frequently increases a crop plant's agricultural, horticultural, or decorative value. Second, transgenic plants can function as living bioreactors, producing commercially essential proteins or metabolites at a low cost. Third, plant genetic transformation is an effective tool for researching gene activity during development and other biological processes. So over 150 distinct plant species, including many crops and forest species, have been genetically changed in over 50 nations globally to far. Plant biotechnology has had a significant influence on plant breeding initiatives because it reduces the 10 to 15 years required to establish a new variety using standard plant breeding processes. Kindly show your support to this channel by subscribing to my channel so that I can make more videos for you. Gene transfer methods in plant. It is now feasible to produce transgenic plants from all main agricultural plant species. A variety of genetic transformations technologies have been established for the creation of transgenic plants. These are mostly classified into two categories. Number 1, gene transfer through vectors, and number 2, vectorless gene transfer or direct gene transfer. First, we will go through vector-mediated or indirect gene transfer. With the help of this diagram, you can see indirect gene transfer method. Agrobacterium mediated gene transfer is the most efficient method of gene transfer because it is a natural method of genetic engineering. The bacterium Agrobacterium tumefaciens causes certain plants to develop a tumor called a gall, which is most commonly located at the crown of the plant, which is the junction of the root and the stem. This crown gall disease affects broadleaf plants including fruit plants, grapes, and ornamental plants. Microbiologists Mary Del Chelton, Eugene W. Nestor, and Milton P. Gordon were the first to demonstrate in the late 1970s that crown gall tumors were caused by DNA from an Agrobacterium plasmid that was transmuted and integrated into the plant DNA. TI plasmid was a term given to the plasmid that caused the tumor. The plasmid is made up of a set of genes, tDNA, that were cut from the plasmid and put into the plant genome. The host plant then keeps the foreign genetic material alive by duplicating it alongside the plant's own gene, which are eventually translated to generate proteins. These proteins are in charge of manufacture of plant hormones, which promote unregulated cell division and the creation of tumors. tDNA also contains genes for the manufacture of metabolites required for the development and replication of Agrobacterium in plant tissue. tDNA boundaries are designated by a particular DNA sequence that may be identified by the enzyme system that cleaves tDNA and inserts it into plant chromosomes, resulting in the creation of crown gall tumor. This natural occurrence of the plant genetic engineering is significant in biology because it shows that DNA may be transmuted between biological kingdoms. Apart from transferring genes across plants, this understanding is currently used to transmit new desirable features from other creatures such as bacteria and animals to plants. As a result, Agrobacterium has emerged as a primary vehicle for plant genetic modification. Let's dive into the mechanism of gene transfer by Agrobacterium. As you can see, TI plasmid tumor-inducing plasmids can be deactivated by deleting the tDNA components while leaving the boundaries intact. Cloning genes of interest in lieu of tDNA allows Agrobacterium to introduce foreign DNA into plant genes, mistaking it for tDNA. The overall method would thus be to create a recombinant plasmid bearing the relevant gene that codes for the desired feature to be transferred to the plant. The gene might be for herbicide resistance or the capacity to produce a particular protein of economic importance such as an antibody or hormone. The recombinant plasmid is then introduced into the Agrobacterium. Finally, bacteria bearing these modified TI plasmids without tDNA are introduced into the host plant where plasmid DNA fragments are inserted into the host plant genome. So this diagram shows the mechanism of Agrobacterium tumefaciens. The first genetically altered food was Favor Savor tomato, and thanks to Agrobacterium gene transfer. Plant tissue such as terminal meristems or leaf disc or cultured plant cells are co-cultured with Agrobacterium tumefaciens containing recombinant plasmids, allowing the bacteria to infect the tissue or cells. Converted tissues are now then put to a shoot-inducing mixture containing 1 or 2 antibiotics that kill bacteria as well as untransformed plant cells. The expression of various reporter genes related with the gene of interest such as glucuronidase, luciferase, or clean fluorescent protein can also detect the transformation. It is to place a root-inducing media after the formation of shoots where it grows roots. After suitable acclimatization or hardening, fully established transgenic plants can be moved to soil. Transgenic plants may be tested for the presence of foreign gene and its correct expression using various molecular methods such as southern hybridization, northern hybridization, PCR, and so on. Number 2. Direct gene transfer, which is physical methods of gene transfer. One of the key issues in Agrobacterium-mediated gene transfer is host specificity as well as difficulties in plant regeneration following gene transfer by infection. Most monocot plants are resistant to Agrobacterium infection. In such circumstances, alternative gene transfer procedures that do not entail infection by any creature have been devised. So here are the direct gene transfer methods. Following are some of the direct gene transfer methods developed for the genetic engineering of specific plants. Number 1. Microinjection. Number 2. Chemical-Mediated gene transfer. Number 3. Electroporation. Number 4. Biologistic method. And number 5. Chloroplast engineering. But before heading ahead, don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my lectures. Number 1. Microinjection. Microinjection is performed utilized automated equipments, robotics, and a microneedle. The desired gene is delivered directly into the plant protoplast as a plasmid or alone. The DNA might be transported straight into the nucleus or into the cytoplasm. Injections into the nucleus are more effective than injections into the cytoplasm. However, microinjections has only resulted in a few transgenic plants. The approach is time-consuming, technically complex, and restricted in terms of the amounts of cells injected. Number 2. Chemical-mediated gene transfer. When treated with membrane-active chemicals such as polyethylene glycol, PEG, and dextrin sulfate, protoplast plant cells without cell walls will take in pure DNA. The DNA gets integrated into the genome once inside the cell and the foreign gene begins to express. This method is heavily reliant on the development of protoplast systems that can regenerate whole plants. Number 3. Electroporation. This is a physical way for delivering DNA into the plant protoplast across membranes using a quick pulse of high voltage direct current. When a high voltage current pulse is administered in the presence of protoplasts, it creates a huge number of temporary holes in the cell membrane. These transient holes aid in the absorption of DNA by protoplast. The DNA is integrated once inside the cell and the foreign gene begins to express. So all these three techniques, chemical-mediated DNA absorption, microinjection, and electroporation, are heavily reliant on the creation of protoplast systems that can regenerate the whole plant. These techniques, particularly electroporation, have resulted in transgenic plants such as maize, rice, and soybeans. However, success rates are modest and the procedures are not always replicable. Number 4. Ballistic method, microprojectile or particle gun method. This is a novel technology that includes employing an electrostatic pulse, air pressure, or gunpowder percussion to propel extremely small particles of stagnant or gold coated with DNA into the cells. The targeted gene's DNA molecules are coated on minuscule gold or stagnant particles. The DNA dissolves and becomes free to integrate into the plant cell genome as a particles transist through the cell. This unlikely procedure really works fairly well, and along with electroporation, this has become one of the preferred techniques. The benefits of BioListic is that it may be used to entire cells in suspension as well as whole or sliced plant tissues. Plant meristems or tissues capable of regeneration, for example, can be directly targeted. This approach, unlike electroporation and microinjection, does not need protoplast or even single-cell separation. Transgenic maize and soybean plants have been created using biolistics. This is a highly mechanical or robotically mediated method as well. Chemical bombardment is often used to create transgenic cereals, and its application has declined as Agrobacterium transformation now works for cereals such as rice and barley. It is still commonly employed in maize and wheat transformation. 5. Chloroplast engineering. Chloroplast includes genes that encode a variety of critical and distinct tasks. Some chloroplast proteins are encoded in nuclear DNA produced in the cell's cytoplasm and then transported into the proper organelle via a specific method. As a result, there are two methods for specific foreign protein to enter the chloroplast. Number 1. A fusion gene encoding the foreign protein and extra amino acids that derive protein transport to organelle may be introduced into the nuclear chromosomal DNA, and the recombinant protein can then be transported into chloroplast following synthesis. Number 2. To change chloroplast function or manufacture foreign proteins, stable genetic transformation of chloroplasts needs insertion of foreign DNA into the chloroplast genome rather than the much larger chromosomal DNA. Kindly give a subscribe to my channel so that I can make more videos in the upcoming week. Applications of transgenic technologies in plants. The introduction of transgenic technologies in plant biotechnology has the potential to outperform past revolutions in production agriculture in terms of technical gains. In 1999, 52% of soya beans, 30% of maize, and 9% of cotton and canola in North America were transgenic. Other transgenic crops being grown include rice, wheat, barley, sorghum, sugarcane, sugar beet, tomato, potato, sunflower, peanuts, papaya, tree species, and horticulture crops. Herbicide tolerance 71% and insect resistance 28%, with just 1% for the other characteristic dominated the commercially cultivated transgenic traits. So here are the application of transgenic technologies in plants. Number 1. Herbicide tolerance. Weed growth is one of the agriculture's most serious concern. These are undesirable that grow alongside with agricultural plants. Because these plants compete with agriculture plants for space, light, water, and nutrient, so crop yield suffers. Herbicides are most often used method for controlling these weeds. However, if the crop is not resistant to herbicide employed, these herbicides might lower agriculture output. Furthermore, pesticides pollute the land and water. Herbicide tolerance in agriculture plants can be achieved by genetic engineering. Herbicide-tolerant crops allow farmers to manage weeds with a specific herbicide while causing no harm to the crop. Engineered resistant to glyphosate, glufosinate, bromocyanide, triazine, 2,4-D, and isoxazoles is now available based on the expression of herbicide in sensitive gene, degradation of herbicide, or overexpression of the herbicide target gene product. The control that transgenic herbicide-resistant crops give to the farmers has resulted in 33% reduction in glyphosate usage on Roundup-ready soybeans and a 20% reduction in glufosinate consumption on Liberty Link canola. Number 2. Insect resistance. Plants are always attacked by many sorts of insects and pests since leaves are their food supply. These pests have potential to harm agricultural plants and diminish output. The use of insecticides to kill insects and pests is a popular pest control method. These substances are potentially hazardous to people and other animals, and they can damage water and the environment. Transgenic plants, by developing insect-resistant crops, provide a viable answer for pest management and environmental production under these conditions. Commercially cultivated transgenic cotton, maize, and potato crops express Bacillus thuringiensis Bt toxin to give resistance to eating insects. Bt synthesis as endotoxin crystalline proteins encoded by Cry genes during sporation. In the alkaline midgut of an insect, prototoxins are cleaved to the active toxin. This binds to the particular receptors in the stomach epithelial cell, causing holes to develop and eventually the insect's death. These Bt proteins or bacteria are not dangerous to humans or other creatures such as flies, butterflies, silkworms, and so on. The Bt cotton plant is most recent example of farming an insect-resistant crop. It is resistant to bollworm Helicobacter armigera, which causes significant damage to cotton agriculture across the world. Aside from cotton, a variety of agricultural plants including potatoes, rice, maize, wheat, tomatoes, soybeans, wheat, ringle, cauliflower, cabbage, and others have been modified with the Bt toxin genes. These transgenic crops have been discovered to be highly successful in pest management, considerably increasing yield while reducing chemical pollutions in the environment. Number 3. Disease resistance. Plants are susceptible to a wide range of illnesses, including pathogenic and deficient disorders. Deficiency illnesses can be controlled by adding to the appropriate manure. Pathogens-caused illnesses, on the other hand, cause crop devastation and financial loss. Fungi, viruses, and bacteria are significant pathogens that cause agricultural yield to decrease or crop damage to occur. This problem can be solved using biotechnological techniques. These are various disease-resistant crops that have been developed to withstand a viral attack. These plants have viral coat protein built into them. This virus-derived resistance has yielded encouraging results in crops such as potatoes, tobacco, tomatoes, and papayas, among others. Some nations have commercialized transgenic papayas that are resistant to ring spot virus. Effective resistance to a wide range of viruses including cucumber mosaic virus, tobacco mosaic virus, papaya ring spot virus, and Cystathenium ring spot virus have been developed. When a plant is attacked by bacterium or fungus, it produces substances such as proteins and enzymes that can combat the invading pathogens. Pathogenesis-related protein or prior proteins are proteins generated in plants as a result of pathogenic attack. They contain enzymes like chitinase as well as antifungal proteins. Phytoxylin are organic chemicals generated in plants as a result of pathogen attack and other damage caused by insect bites. A significant range of transgenic crops with improved resistance to different fungal infections have been developed with genes responsible for manufacturing any of the antifungal chemical listed above.
Number 4. Abiotic stress tolerance. Abiotic stress is another important component
Affects plant development and production. Plants create metabolites in response to stressors such as excessive temperature, drought, or salt. Among these metabolites are stress-related osmolytes such as sugars, sugar derivatives such as trihalose, fructans, mannitol, etc., amino acids such as proline, glycine, betaine, heat shock proteins, and antifreeze proteins. Crop plants have been genetically modified to overexpress genes involved in the synthesis of one or more of the metabolites listed above. Various research organizations have created salinity-tolerant rice and drought-resistant sugarcane. Number 5. Delayed ripening fruits and vegetables. One of the issues affecting agro-industries is the shelf life of fruits and vegetables. A variety of major metabolic pathways contribute to fruit development and ripening. One of the main molecules involved in fruit ripening is the gaseous hormone ethylene. The process of fruit ripening can be sped up by inhibiting ethylene production. Commercially available methods for extending shelf life includes switching off polygalactoronase genes, switching off genes in the ethylene biosynthesis pathway or degrading intermediates in that pathway, and expression of cytokinin genes. Antisense messenger RNA technology is employed for this purpose. In the instance of antisense messenger RNA technology, a reverse copy of a gene, a complementary gene, is inserted in a plant with all of the required regulatory components present in the regular gene, for example, antisense messenger RNA for ethylene biosynthesis gene. Because ethylene production may not occur in such plants, fruit ripening will be excessively sluggish under normal conditions. This delayed ripening aids in the export of sensitive fruits like tomatoes. Flavor Saver tomato is a superb example of antisense technology. It was debuted in 1994. Ripe tomatoes generally release the enzyme polygalactoronase, which digests pectins, weakening the fruit and making it more prone to fungal diseases. Scientists extract the PG gene, which creates a complementary gene that produces a complementary messenger RNA that binds to the normal messenger RNA and inactivates the normal messenger RNA for this enzyme. Kindly don't forget to press the subscribe button. Number 6. Male sterility. Male sterility is an essential feature in plant breeding because it limits the self-pollination and encourages cross-pollination. As a result, creating male sterility in agricultural plant is critical in terms of plant breeding. Male sterility is introduced in agricultural plants by introducing the gene code for the RNA hydrolyzing enzyme burnase. This enzyme prevents pollen production, rendering the plant male infertile. With the aid of the tapetal specific promoter TA29, this gene is engineered to express solely in the tapetal cells of the anther, limiting enzyme activity to the cells engaged in pollen generation. The insertion of another gene, BAR*, which inhibits or suppresses the function of burnase, can restore male fertility. Number 7. Transgenic plants with improved nutrient qualities. Crop plants can be genetically engineered for the improvement of nutritive qualities such as the edible parts like grain, fruits, tuber, etc., protein content, starch qualities, etc. So here we will discuss golden rice. Rice is often devoid of vitamin A. Golden rice, a genetically altered rice, does produce vitamin A. A lack of vitamin A can lead to disease known as night blindness. In the third world nations, this problem is severe. The goal is that introducing golden rice would rescue millions of people from the repercussion of vitamin A deficiency in nations where rice is the mainstay of the diet. Protein quality of seeds and tubers. Nutritionally enhanced food will have more nutrients such as proteins, vitamins, and other beneficial compounds. Protein molecules rich in essential amino acids can have their genes extracted and transplanted to food crops with appropriate promoters to express them in edible section of the plant. Other examples of boosting the nutritional content of food materials by genetic engineering includes protein-enhanced sweet potatoes, rice, wheat, and maize, high vitamin A canola oil, and improved antioxidant fruits and vegetables. Number 8. Transgenic plants as bioreactors. Molecular farming. Modern techniques for producing transgenic plants and isolating new genes responsible for the manufacture of numerous industrial proteins, enzymes, and medications for human and other species have resulted in the creation of a whole new and remarkable technology. Even many animal proteins and medicines can be grown on the farm. Plants are very cheap, and the raw materials necessary for plant cultivation, harvesting, and product extraction are quite easy and inexpensive. If the plants are transformed with the appropriate gene and promoters, they can serve as bioreactors to create the essential chemicals. In adequate amounts such as proteins, therapeutic proteins, antibodies, vaccinations, enzymes, amino acids, vitamins, medicines, and so on. Some examples are diagnostic and therapeutic proteins. By using plants as a bioreactor against the use of conventional fermentation techniques, which include costly equipments, upstream and downstream processing, the cost of the production can be brought down substantially. Proteins such as plasma protein, peptide hormones, interferons, and cytokinin can be produced in specific organs of transgenic plants such as the leaf in the case of tobacco, tubers in the case of potatoes, and stem in case of sugarcane. A recombinant form of natural hormone. RBTS, bovine somatotropin, which causes cows to produce milk, can be synthesized through transgenic plants. RBTS increases milk production by as much as 10 to 15%. It can be used to treat over 30% of US cows. Food enzymes, including a purer, more stable form of chymosin used to curdle milk in cheese production, are now being synthesized by transgenic plants. It is used to make 60% of hard cheeses. This can replace the chymosin of rennet from slaughtered calves' stomachs. Edible vaccines. Transgenic plants can be also utilized as edible vaccinations if the vaccines are expressed directly in the plant's edible components. If the plants are transformed with the vaccine-producing gene linked to an organ-specific promoter, it will express and the raw edible component may be consumed for immunization. The vaccine molecules in raw food, tuber, fruits, or leaves will be absorbed by the cell lining of the mouth and food canal as it travels to the stomach. This sort of vaccine may be used on both people and animals. For example, fodder plants can be genetically modified to generate the productive antigen Agrobacterium, allowing calves to automatically immune against the deadly anthrax. Antigenic protein production in edible sections of the crops such as tomatoes, bananas, and apples is essential for human vaccination. Without refrigeration, edible vaccines are simple to store and carry. The delivery technique is also quite straightforward. The cost of the product is relatively inexpensive since expensive equipment and downstream processing procedures are not required for industrial fermentation product. Vaccination against illnesses such as chlorella, hepatitis, smallpox, TB, and others will also be in reality in the near future. Number 9. Biodegradable plastics. Because plastic is not biodegradable, it causes a swirl of environmental issues. Some people may generate biodegradable polymers such as polyhydroxy butyrate (PHB). In Arabidopsis, plants are genetically altered with the gene responsible for PHB biosynthesis. They manufacture these PHB globules in the chloroplasts. This bioplastic is easily recovered and purified from plants' leaves. A number of firms are already creating transgenic plants to investigate the feasibility of generating biodegradable polymers on a large scale. Number 10. Plant secondary products. It is feasible to use metabolic engineering to improve a specific secondary metabolites or to develop a novel chemical biomedicinal plant. In general, overexpression of a key enzyme gene in the biosynthetic pathway results in increased synthesis of desired product. For example, metabolic engineering approaches in Texas Pecuta tissue cultures have significantly increased the production of the anti-cancer medicine Taxol. Induction of hairy roots cultures with Agrobacterium rhizogenes is another in vitro approach for increasing the synthesis of metabolites, particularly those generated in the roots. Root cultures may provide as a source of root-specific compounds. Kindly don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my lectures. Risk associated with genetically modified food. The use of genetically modified plants and animals have become commonplace in today's society without many people being aware of it. The lack of consumer consent in the choice of eating genetically modified food creates an ethical dilemma. Much as 70% of the food prepackaged in the normal grocery store contains genetically modified foods. So the question arises, are they all safe? Flavor saver tomato, genetically modified tomato that rot longer but less resistant to pests, easily caught diseases, which makes the crop even more costly to grow and thus higher price for consumers. Food allergies. The biggest issue regarding GM crops is their proclivity to cause allergic responses. The dread of allergic reaction originates from the belief that any protein might cause an allergic response. Companies introduce a new protein into an organism when they create a new gene, and it is uncertain whether or not these new proteins may trigger allergic responses. Brazil nuts and soybeans are two examples of allergy threats posed by genetically modified foods. To make soybeans more nutritious, a protein from Brazil nuts was added. However, it was discovered that those who were sensitive to Brazil nuts were also extremely allergic to the GM soybeans. Creation of superweeds. Another issue arises by genetic engineering is the development of superweeds by horizontal gene transfer from GM crops to wheat crops. Crop hybridization with neighboring weeds may allow weeds to acquire features we don't want them to have, such as herbicide resistance. Similarly, genes for viral disease resistance or insect resistance might benefit wheat production near a crop field. Creation of superbugs. Antibiotic resistance is combined with new genes while creating GM crops, since gene transfer is only effective in few cells and a marker is required to detect successful transfers. The risk of gene transfer stems from the possibility that genes from GM crops would be picked up by harmful bacteria in people's guts. If bacteria containing antibiotic resistant genes create illnesses, doctors may find it difficult to cure. Environmental impacts. Environmental risks involved with GM crops are numerous. The number one is decrease in the biodiversity. They include the capacity of the genetically modified organism to escape and perhaps introduce the modified genes into natural populations. The most serious issue is the possible loss of biodiversity in crops and their accompanying insects and animals, which might change the forces of natural selection due to direct human interference. Number 2. GM pesticide producing crops. These are hazardous to non-target species. For example, long-term exposure to pollen from GM insect-resistant maize affects the behavior and survival of America's most famous butterfly, the monarch butterfly. Number 3. GM crops are toxic to beneficial insects. For example, GM Bt crops adversely affect beneficial insects important to controlling maize pests such as green lacewings. The toxin Cry1AB affects the learning performance of honeybees. Number 4. GM crops are a threat to soil ecosystems. Many BT crops secrete their toxins from their roots into the soil. Residue left in the field contains the active BT toxin. Generally, GM herbicide-tolerant crops are connected with one of the two herbicides, glyphosate or glufosinate. Both herbicides are problematic. Several new research indicate that Roundup is significantly more dangerous than previously considered. It is poisonous to aquatic species such as frog larvae, for example. Number 5. Poisoned wildlife. The introduction of new genes into plants might have catastrophic effects for animals in a variety of situations. Engineering crop plants such as tobacco or rice to create polymers or medicines, for example, might imperil mice or deer that devour crops or crop waste left in fields after harvesting. Let's go into the oversight. To maintain the balance between the benefits and hazards of genetically modified foods, adequate supervision is required to keep the risks indicated above in mind. Many measures are taken while analyzing the safety of novel GM foods to determine the safety of new proteins. In the United States, 3 major entities are active in the regulation of genetically modified foods. Number 1, the Food and Drug Administration (FDA). Number 2, the Environmental Protection Agency (EPA). And Number 3, the United States Department of Agriculture (USDA). So concluding it all, there are various possible advantages and concerns associated with genetically modified crops. Several safety investigations have had both favorable and poor outcome, and long-term research to assess safety and dangers are also required. Thank you so much for watching till the end. Don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my videos. Kindly show your support to this channel so that I can grow my audience. Thank you so much.